So I will be talking about eDNA sampling and analysis for biodiversity assessment and conservation. And eDNA is a relatively recent new tool in the molecular toolbox, and as such is providing new opportunities with exciting prospects for conservation studies. And in the past 10 years or so, eDNA applications have really burst, expanding from the initial applications that were mostly focusing on species detection to now being able to assess biodiversity and characterizing communities at the ecosystem level, and also allowing us to reconstruct trophic interactions. And more recently, eDNA can also be applied to give us insights to intraspecific variation. And this is something very, very recent. So given this breadth, these broad applications today, in my presentation, I will mostly focus on applications and give some examples concerning mostly wildlife studies. And I want to start by spelling out what eDNA is. eDNA is the DNA that can be extracted from the environment, taking samples from the environment, such as soil, water, ice, snow, air, and so on. So the key thing here is that we don't need to first isolate any organism or not even any part of the organism of interest. And this is possible because every organism shed naturally or release naturally DNA in the environment through biological, physiological processes. And when we work on wildlife, and we've heard about it in the previous presentations, in terms of sampling, we are used to distinguish between invasive and non-invasive sampling, with invasive sampling requiring handling or capturing the animals. And then there is all a variety of non-invasive techniques that allow us to minimize the impact on the animals. So being able to sample DNA from the environment greatly expands our ability to non-invasively study wildlife. And you'll notice here that under the different kind of eDNA samples, I also listed a scat sample, the same scat sample that is here under non-invasive genetics. But the difference is that here we target, instead of that the DNA of the animal that left the sample, here we target the mixture of DNA of all the different food items that the animal have potentially eaten. And so how does eDNA work in practice? The workflow, it's in general lines similar to other molecular techniques. So we go out in the field, we collect an environmental sample, and we bring it back in the lab where we extract the DNA. And then we use different molecular approaches depending on the question that we have. We may use single species approaches when we are interested in a known species of interest, or we may use multi-species approaches, also known as DNA metabarcoding, that allow us to target in the same experiment, in a single sample, the many species at the same time, or approaches that allow to discriminate between individuals. And then we get DNA sequences, and these are informative and are used to identify the species or the many species that are comprising a sample, or to distinguish between the various individuals of a species. And then these data are used for the subsequent ecological genetic data analysis. So this workflow in general, it can look similar to other eDNA methods, but what we can do now with eDNA, what made it possible to work with eDNA now, it's really the impressive technological revolution that had occurred in the past 10, actually 15 or 20 years that brought improvements, significant improvements at all these stages of the process. So I will give now some examples, starting from the simple detection of single species to the detection of many species to describe biodiversity in ecosystems. So the earlier applications, as I said, concerned mostly the detection of single species. And here I'm giving the example of one of the first seminal studies. So back in 2008, when researchers showed that by sampling water for ponds, they were able to detect an invasive species, the American bullfrog, that has been introduced globally and that is now responsible for the decline of several endemic amphibians. So in they succeeded in demonstrating that they were able to detect this species in several ponds that were known for the presence of the invasive species. This way, they showed the potential of the method for tracing the spread of the invasion, but also for detecting other possibly elusive cryptic species, including endangered species, and therefore estimating their distribution, and also study cryptic aspects of their biology, like spawning behavior, for example. Then, with the development of DNA metabarcoding, the multi-species approach, eDNA methods were soon used to describe biodiversity in aquatic ecosystems, in the beginning, especially in freshwater systems. And these studies showed that the eDNA method, the green bar in these graphs, outperformed the traditional biodiversity surveying methods, the blue bar in these graphs, in terms of the number of species that were detected. And the taxa were also detected with a higher taxonomic resolution and in a very cost-effective way, so with less effort compared to the traditional methods. These earlier studies also soon showed that still, the best picture of the biodiversity in an area was provided by the combined application of the eDNA method with the traditional method. Today, we use eDNA for biodiversity surveys in both aquatic and terrestrial ecosystems with a variety of sample sources, and to describe species assemblages, monitor biodiversity in space and time, and also the health of the ecosystem, because we can also detect parasites or agent of diseases. And we can also evaluate, for example, the impact on biodiversity of anthropogenic actions or of restoration policies. Also, another use of this method is to get insight into past biodiversity, for example, using samples that refer to past environments, like here in this figure, permafrost soil. And now I will move on to different kind of application that concern the like being able with eDNA to obtain the list of species comprising an animal diet for going up to the construction of complex food webs. And so here the concept is the same, except that the environmental sample that we're starting from is a scat sample that may contain DNA of plants and animals, depending on the diet of the predator that we're studying. Here we use the multi-species approach, the DNA metabarcoding, to reveal the different species comprising or composing the fecal sample collected. And we used this approach in a study of wolf diet in Croatia, together with the WWF. This study is now completed, and now with WWF, we are also working on brown bear diet study. So for this particular wolf study, we used scat samples that were collected in different areas of Croatia. And these scat samples were originally sampled and were already processed in our lab for individual identification. And we then used them for diet analysis. So this is also to emphasize the many life that or uses that a scat sample can have, so it can be used for multiple purposes. And we used the DNA metabarcoding approach that I just explained, and we were able to reconstruct the diet composition for the population, showing that most of the sequences detected in the diet belonged to members of the Cervidae and Suidae family, with the red deer, roe deer, and the wild boar being the species most frequently found in the samples. And by looking at the samples individually, we also saw that most of the samples were comprised by a single diet item, as you could see by the different colors in this graph. But the interesting thing was also when we looked at the spatial distribution of the samples with their diet composition, because you could see that some spatial patterns are apparent, with the deer, the green and the blue color, being found mostly in samples collected in the northern part of the study area. And while the diet seems to transition more towards a higher consumption of wild boar and also of species possibly associated to anthropogenic activity here in the central part of the study area. And this is consistent with what we know about prey availability and also the distribution of human presence and activities in the study area. And it seems to suggest that the wolves shift their diet based on the availability of their preferred prey. And this is also consistent with another similar study that was done in North America, where they also looked at the spatial patterns in the diet using eDNA metabarcoding. But in this case, they went a little bit farther than what we did, because they had more samples, and also they had ecological data in relation to the samples. So they have a lot of metadata for the samples. So they looked at, for example, different the diet, a different feeding behavior between the denning and the annual home ranges, and then differences between pups and adults. This is just to give an example of how the detailed information in eDNA metabarcoding diet data could be used to address questions about feeding behavior, perhaps in relation to sex or age classes, or differences in anthropogenic pressure in different study areas. When more samples, when DNA metabarcoding data can be obtained for more samples in association with relevant metadata. And in these cases, when we have all of this information for many samples, we can even go a step up. We can start constructing food webs that reflect energy flow between and through ecosystems. We can start by constructing simple networks, simple food webs, like this one that I've worked on with a student of mine for brown bears in the Tatra Mountains in Poland, where we were interested in the trophic interactions for the bears in this area. And this is a simple bipartite network where down here we have the scat samples that were collected across the ear for the brown bear. And down here where there are the different food items that were identified using DNA metabarcoding. And we are highlighting here the trophic interactions with a key species that we're interested in, the bilberry, which is a species of economic interest. So we use this result to emphasize the role of the brown bear in providing an important ecosystem service, which is the seed dispersal of these important species. So when we can construct networks or like this for several predator species, then we can build more complex food webs, like this one, for example, that was constructed using eDNA metabarcoding diet data for 2 carnivores, 2 omnivores, and 2 herbivores. And you could see how with something like this, we can start to have more insights into the community structure. And again, this is possible because eDNA metabarcoding, when applied on diet, offers a lot of detailed information, starting from a higher species detection with high taxonomic resolution, and also in a very cost-effective way that allow us to analyze more samples at the same time. So far, I mainly talked about examples that related to focusing on species detection in ecosystems or in animal diet. But as I mentioned before, more recently, DNA metabarcoding is also allowing us, eDNA methods are allowing us also to get insights into intraspecific variation in populations and also in individuals. This part of the field is really still in its infancy. So I will just give some brief example just to give you the taste of what could be potential development. And I'm starting with what researchers are now doing or can now do, especially in marine environment, exploiting the aggregating behavior or many marine megafauna. Like in this picture, I have an aggregation of whale sharks, but there are actually many marine mammals that also move in groups through in the water. And all these animals shed skin cells naturally when moving through the water body. So researchers here collected water from the middle of the aggregation, and from the eDNA extracted from the water, they were able to identify sequence variants corresponding for this aggregation. These sequence variants partly matched other variants or other sequences that were already published using independent DNA data. And in some cases, they also found new sequence variants. This data can be used, like in these figures, to map these variants and kind of allow us to look at the geographic distribution and also at possible genetic structure for the population, or can also be used to build networks like this one can be used in population genetic studies. So this is an important step forward to study marine biodiversity, particularly a lot of these marine mammals that are typically hard to study using traditional methods, and some of which are also of conservation concern.
Marta, only 2 minutes more.
Thanks. Only even more recent achievement is individual genotyping from eDNA. This was possible, is possible for now only in terrestrial ecosystems, because here there is less eDNA mixing or dilution compared to water bodies. And it's now been possible in snow, using snow tracks, because snow act as a natural freezer, therefore helping the preservation of eDNA. Researchers have been trying to achieve individual genotyping for a long time, but we were able already more than 10 years ago to already do species ID from snow, but probably the methodology were not there yet to allow us to get to individual ID. But surprisingly, now, just last year, three different research groups independently, including Melanie group that will talk extensively about these after my presentation, and also my group in Ljubljana, were able to arrive to these important achievements, sort of indicating how probably the technologies or the methods were not there yet, and then the improvements that we obtained or that were made during this time now allow us to do what it was not possible to do just a few years ago. So Melanie will talk extensively about this in the next presentation. I just wanted to mention very quickly what we've done in our research group. We were able to genotype at the individual level snow track eDNA from 3 large carnivores. To do this, we used an approach based on the new high throughput sequencing technologies, which we believe gave us greater power to retrieve and analyze the low quantity DNA from snow track samples. We observed differences in the success rates between the species, and we think that there is probably, therefore, an effect of the ecology of the species in affecting our ability to retrieve and analyze eDNA from snow track. And this is something that we'll have to take into account for a broader application of the method. But these represent a significant step forward for complementing current non-invasive genetic methods. And I will briefly conclude just restating and bringing forward again the new opportunities and advantages that eDNA methods can offer to study biodiversity and wildlife conservation in particular. I will not repeat them because I've already mentioned and discussed them over the presentation. But since new opportunities come also with new challenges, I just want to briefly mention some of the current limitations of the method that are mostly related to, first of all, our limited understanding of what we call the ecology of the eDNA, which is the combination of all the different aspects that relate to how eDNA originates, the state that it's found when it's released, the fact that it moves through the environment. So we take a sample here, but maybe eDNA originated somewhere in another part of the ecosystem, and the fact that once out there, it degrades rapidly. And then there are methodological bias and bias related to the taxonomic assignment and to the interpretation of the quantitative signal. The good news is that we are already learning a lot on how to deal with this bias, and maybe these limitations may be, or at least part of them may be, overcome as we progress in our learning. And the important thing is to take account of this possible bias throughout the entire eDNA workflow through careful study design. And with this, I'll thank you for your attention and acknowledge and thank all the supporters, including some of our collaborators on some of this work. Thanks.